And that brings us to microfauna. Microfauna, we're going to talk about 4 different groups: nematodes, rotifers, tardigrades, and protozoa. So nematodes, rotifers, and tardigrades are eukaryotes. Protozoa are prokaryotes. So I've got pictures here of a little roundworm or nematode, a adorable tardigrade that's standing up and looking at us, a rotifer where you can see the wheels on its side, and then down in the bottom right with that red dot is a stentor, which is a kind of ciliate protozoa. So let's talk about it. Next up, nematodes. For those who haven't taken invertebrate zoology, nematodes are unsegmented roundworms. Most of them are microscopic. The soil ones are usually fairly microscopic, but they are very, very common. My invertebrate zoology professor used to say if you could put on nematode goggles, or nematode goggles if you're British, and see nothing but nematodes, you'd still see the world, because you'd still see all the animals walking around because they're so full of nematodes. You'd still see all of the geological features because there's so many in the soil. Nematodes are everywhere and they're super plentiful. It's one of the more common things in our soil. They specifically have to live in the water-filled pores of soil. They have soft body tissues. They need the moisture. But they have a really cool thing where if the soil dries up, they can coil up into a cryptobiotic state where they are pretty much impervious to anything. They don't need any oxygen for respiration, and they can survive some extended dry periods. The majority of soil nematodes are feeding on things that are smaller than them: fungus, bacteria, algae. There are some that are parasitic. So this here is, these little nodules are parasitic colonies of parasitic nematodes. They can be problematic for farmers. Parasitic nematodes can infest several root vegetables, but they aren't harmful to humans. They also stimulate nitrogen release in the soil because they eat a lot of bacteria, and bacteria are one of the few organisms that can fix nitrogen. So we'll talk in a later lecture about nitrogen fixing, but the bacteria, or the nematode, doesn't need as much nitrogen as is in the bacteria. So by eating those nitrogen fixing bacteria, the nematode is releasing that excess nitrogen into the environment. Brings us to rotifers and tardigrades. So these are still both eukaryotes. They are multiple celled complex structures, but they're both quite small. So this picture shows a one-celled paramecium, which is a protist, that is almost as large as the many-celled rotifer sitting beside it. So depending on the protist, they can eat small rotifers. Rotifers typically feed on dead things. They're detrivores, they're filter feeders, but they'll eat anything that gets close to their mouth, anything that's small enough. So that means they're normally eating things like bacteria and tiny, tiny protists and dead things. Tardigrades, on the other hand, are usually saprophytic. They're sucking the fluids out of algae and plants. And I'll show you pictures of tardigrades, not the next slide, but the one after. So here you have our rotifers again. You can see in these pictures those circles on what I'm going to call its head. The very first scientists saw those and looked at them as wheels. So rota, reel, fera, to bear. They are bearing wheels. They have these wheels on them, which are really important as they are detrivorous filter feeders. So they're spinning those, getting a current, pulling anything that's small enough really into their mouths. So they need to be in water. So when you find them in soil, you find them in wet soil, you find them in the pore spaces between the soil granules. When you find them in aquatic systems, they are big filter feeders in terms of like cleaning the water. People will put them in their fish tank to like clean that water. They can be sessile, just sitting there or floating there, and they can be mobile. They'll like, especially soil rotifers, will move in like an inchworm style fashion. Tardigrades, tardigrada, slow stepper, because they walk. They have the lumbering walk, which is also how they got the other name is water bear. As I said earlier, they have this like tubular snout that they use to suck the liquids out of plants and mosses and things. So you typically find them in mossy soils and wetlands. You're not usually finding them in forest soils, but sometimes in forest soils. I love this beautiful, the lower picture is this beautiful electron scanning microscope where you can see the little claws on their feet and their tubular mouth. But when I find them under a microscope, it's more like the top picture, where it's just this little knobby oval. And if we do see them, this is what we'll end up seeing. All right, so let's move on to the prokaryotes. We have Protozoa. I'm going to use some new terms and some old terms because Protozoa are one of those, I'm going to say family, but it's not family, groups of organisms where our knowledge of Protozoa has increased drastically in a short time, faster than the English vocabulary can keep up. So, for ease of speaking and classifying, I'm going to group all of the soil protozoa into three groups: amoebas, ciliates, and flagellates. They are all found in plankton, so they're generally aquatic. They need to live in the water-filled pores. They have soft body tissues. They will dry out very fast. Some of them can form resistant cysts, where if it's too dry, they'll form this cyst where they can withstand dehydration and then come out of that sort of torpor when there is water again. They're mostly feeding on bacteria because it's the only thing that's smaller than them, really. And because of that, they're another one of those organisms that produces a lot of useful nitrogen. As we said before, I'll talk more about nitrogen. Even though 60% of our atmosphere is nitrogen or more than that, plants can't use most of it. So they require nitrogen to be fixed. And we'll talk about that later. But that's one of the reasons Protozoa is important in the soil. So the first of these groups are amoebas. Right now there's a phylum of Amoebozoa, but traditionally we think of amoebas as any planktonic organism that has pseudopods. And we can picture these sort of soft body cytoplasmic arms sort of stretching out. But not all amoebas look like that. A lot of amoeba's pseudopods are axiopoda, so they're like spiky. Some have shells, so they're called testate. So this little guy down here is the same type of amoeba with those cytoplasmic arms that in the first green picture, but now he's got a shell and he just sticks out his arms when he wants to and pulls them in when he wants to. We can have parasitic amoeba. Some can be found in the soil, some can be found in water. You hear about them in the news, parasitic amoebas in people's brains. They're rare. Less rare are slime molds. So this is a sort of newer classification. We didn't used to include slime molds in the phylum Amoebozoa, but more recently we've started to include them. Before we thought of them as a fungus because they sort of grow like a fungus in the soil. Slime molds are really, really cool. And I could probably teach a whole lesson just on slime molds. They're really neat, but you'll usually find them in fungus books because we've traditionally thought of them as funguses, as molds, not as amoebas. The next ones are flagellates. So they don't have their own phylum. These are any of the prokaryotes and some eukaryotes that have whip-like appendages that they use for locomotion. So most of these are planktonic prokaryotes like Euglena. This is probably the most common one. If you took first year bio with Joe and Laura, you've probably seen Euglena. They're a pretty common flagellate. The other famous one are dinoflagellates. They're found in aquatic environments typically. They're really famous for algae blooms, toxic algae blooms is where we get dinoflagellates. We do have some parasitic flagellates as well, typically not found in soils, more likely found in aquatic environments, but I like to bring them up because I spend my summers canoe tripping and Giardia or beaver fever is a very problematic flagellate that is common. And our last group are ciliates. So there is a phylum called Ciliophora. I'm not sure if it includes all ciliates or how that works, but the most common ciliates are part of the current phylum Ciliophora. I've got a picture of a paramecium here, which is probably the most well-known ciliate. So they have organelles that look like little hairs. You can see them in this picture. They frequently have multiple nuclei, so they'll have micro and macronuclei. It's very common of this phylum. And most of them are heterotrophs, so they're going out and eating things. And again, they're eating things that are smaller than them, so a lot of algae bacteria, but as we discussed earlier, there are whatever they can fit, they will eat. The cilia are usually used for locomotion, but sometimes they use them to feed. So here's a beautiful picture of a stentor. Stentor are sessile, and they use their cilia to bring in food like a rotifer does.